Hi everybody, my name is Gabriel Ohlsson and I'm going to talk about getting the bigger picture by analyzing surface interactions at the nanoscale level. You can see me to the right in this picture. I work at a company called Biolin Scientific. We are a scientific instrumentation company that provides different kinds of surface analysis instruments. We have three main brands: Attention, which is our surface tensiometer brand; Case-Venema, which is our range of Langmuir-Blodgett instrumentation; and then we also have QSense, which is our quartz crystal microbalance with dissipation monitoring instruments. So this is what I will talk a little bit more about today. We have our headquarters in Sweden, and we have sales offices and distribution partners worldwide. So small causes can have large effects. This picture here, it kind of refers to the butterfly effect. It's a movie from the early 2000s. And the idea is that like small events somewhere can have a big impact somewhere else. And this is true for many things, actually. If you are, for example, infected by a virus, it starts at the nanoscale level, at the surface, at the cell membrane, one virus infecting you. And eventually this will spread and it will make you feel sick. If you want to treat or cure the disease, you want and need to attack the virus also on the nanoscale in order to have a macroscopic effect eventually. So there are many events that take place and originate from nanoscale interactions at surfaces. So this is why we have developed this QSense brand and QSense technology. The idea is to provide a surface sensitive technology that can give you information about changes in different kind of interaction processes taking place on surfaces. So typically you can measure mass, thickness, viscoelastic properties in a label-free manner, and time resolves. It's a real-time measurement. So with this kind of technology, here we have a few examples of what you can do. And the pictures to the right illustrate some typical situations or systems that you can measure and get information about. So the yellow bottom is kind of the surface of the sensor. The sensor is typically operating in a liquid environment, and you introduce your liquid sample material to the sensor surface. So in the top here, we can see some molecules interacting with the surface. First, layer of green molecules, followed by a layer of red molecules. So we have different adsorption and binding events that we can measure in real time. Second example here, we have binding event that causes some kind of conformational change. And in the third case, we are looking at cross-linking of polymers. And next we also can follow and study swelling of polymers. And eventually, the last example here, we have degradation of molecular layers from surfaces. So these are some typical examples of the processes that you can study. One relevant question is, of course, about the sensitivity and what kind of size range are we talking about here. This instrument, the quartz crystal microbalance, is it can detect really small masses down to less than half a nanogram per square centimeter. So it means that you can measure the interaction or binding of really small molecules on a surface. So we're looking at like sub-nanometer, angstrom level of size. And then bigger molecules like surfactants, protein, DNA, micelles, nanoparticles, up to the micron range where you can find live cells. This is how the instrument typically look like. Everything is based on the quartz disc that you can see here. It's 14 millimeter in diameter. quartz sensor, and this is mounted in a flow module. This is made out of titanium, into which you can introduce the sample material so that it will interact with the surface. And the flow module is placed in this kind of chamber platform, which is temperature controlled. You can go from close to 0 degrees up to 150 degrees Celsius, depending on the setup that you're using. And then you, of course, you connect it to some kind of pump or liquid system like this. And this instrument, it can be used during different processes and different stages of your research. First of all, if you just want to have some more basic understanding of a system, what is happening, what kind of process do I get, how does this new molecule interact with that particular surface, for example. Secondly, you can characterize and quantify to see how much of a certain molecule attaches to a surface, how fast does it go. You can also optimize to see and find what are the best conditions for the particular system that you're looking at. Maybe you want to optimize temperature, pH, salt concentration, or something like that. So let me show you a little bit how it looks like when you have performed an experiment, what you can really see. Here we have 3 examples of basically quantification. In the first left picture here, you can see a binding of small molecules onto the surface, and you can follow in real time the mass change like this. And eventually we have some release as well, and you can see that the mass goes down. So you get some kind of profile with the binding rate and release rate like this. In the second example, we have a buildup of polymer layers, first layer and second layer, so we can follow the thickness as a function of time. And the last example here, we see and follow the softness or the viscoelastic properties as a function of time, going from more rigid to more soft to more rigid again, swelling and collapse of polymer layers. And here I also would like to illustrate that you can get the rate, the adsorption rate, desorption rate, extension or swelling rate, and cross-linking rate as well, since it's time resolved, real-time measurement that you're having here. Okay, so as I mentioned, everything is based on quartz, the quartz disk, the quartz crystal microbalance. And the quartz sensor itself is clamped between 2 gold electrodes, on top of which you can put any kind of material, of course, to mimic the situation you're interested in. Quartz is piezoelectric, and that means that if you are applying a voltage to the quartz, it will deform, and vice versa. If you deform it, it will generate the potential. If you apply an alternating voltage with a certain frequency, you can make it oscillate at its resonance frequency. In our case here, we have a 5 megahertz resonance frequency. And the resonance frequency or change in resonance frequency scales or correlates to the mass load of the sensor. The second parameter that you measure is the dampening or the dissipation of this oscillation motion. So if you have a more soft material on top of the sensor, the dampening will be higher compared to if you have a more rigid material. So let me show you how the raw data would look like. So here we can follow changes in the blue curve, resonance frequency, red curve, dissipation. Start with a bare sensor in just a running buffer to establish a stable baseline, and then we measure the relative change in frequency and dissipation. First, we have an adsorption of red molecules. They form a relatively thin and rigid layer. We have a certain frequency change correlating to the mass change. Dissipation increases just a little bit because we have a rigid case here. And the second layer, the green molecules, they are bigger, heavier, forming a more soft layer. So the frequency shift is bigger and dissipation is also bigger. because of the larger mass and increased softness. So this is the working principle, how the machine actually gathers the data, and this can then be translated by the data analysis software into mass, thickness, viscosity, elastic shear modulus. Let me show you a few examples how you can use this in the field of developing new pharmaceutical drugs, or if you want to target, for example, the problems that we see today with the COVID virus, we have the Zika virus and other different new diseases that are coming up. We have a demand for new pharmaceuticals, new drugs, vaccines, and in order to prepare and develop these faster, we need to have powerful tools for that as well. First of all, I would like to talk about lipid-based systems. You know that lipid membranes, they are present everywhere in living organisms. And these are the shells that surround the cell, for example. So if you would like to access the cell, you need to go through the membrane in some way. For example, if you want to cure or treat a sick cell, you need to target that cell with the drug molecule. And also, of course, if the virus is infecting you, it will also, first of all, bind on the surface or the membrane of the cell. This has been used and studied quite a lot with QCM-D, since you in real time can measure the conformational changes and changes in mass and thickness of layers on a surface. This is an excellent tool if you want to study the formation of model membranes that are used then for different kind of development or research around new pharmaceutical drugs. Here we can see in the example here, we have lipid vesicles adsorbing to a surface. They rupture and form a bilayer, which is basically the cell membrane or model cell membrane. And then we have attachment of some molecules to the receptor proteins that are integrated into this bilayer. And then you can follow with the frequency and the dissipation, the attachment of the vesicle, the rupture of the vesicles, and the binding of these molecules. So you can get information about thickness as a function of time, viscosity, and elastic shear modulus. This is a nice and powerful tool to evaluate the membrane quality and formation of the lipid membrane. So let me give you a more concrete example how this can be done. So here we are using a model membrane platform to evaluate nanomedicine uptake and release of the drug molecule from the nanoparticle carrier. So in this case, we are using drug-loaded nanoparticles. These are really interesting candidates for, drug delivery, because you can create this kind of targeted drug delivery vessels. For example, in cancer treatment, it's more favorable if the cancer medicine directs or targets only the sick cells and not the healthy ones. That will reduce the suffering a lot for the patient and make it much more efficient, the treatment. So here we're interested in some basic questions. Basically, if you can adsorb these nanoparticles to the cell membrane, and we want to see how they arrange and if we can release the drug in a controlled manner as well. So here we have basically different scenarios that we would like to evaluate and see if it happens. First of all, we form the membrane on the sensor surface. We introduce the nanoparticle drugs. We can see if they attach more evenly distributed, if they collapse or aggregate on the surface. And then how is the drug released? Do we have a complete release or partial release? The experimental design for this is that we have cell membrane mimic that we form. We have 3 different nanoparticles, non-pegylated, pegylated, and partially pegylated with different degree of positive charge. And then we have a reducing agent in order to disintegrate the nanoparticle and release the drug when it has reached the cell membrane. So this is what we do. We compare and analyze these 3 nanoparticle candidates to see how they behave during the adsorption and the release process. So here we have some data. What we can see here, first of all, we have the formation of the lipid membrane on the surface, and then we have the 3 different nanoparticles and adsorption characteristics thereof. We can see that the non-pegylated, we have more of them attaching to the surface compared to the pegylated, we have less of those attaching to the surface. When we rinse, we see that we lose some of the nanoparticles from the surface because we have this increase in the frequency. And then during addition of the reducing agent, also they dissolve and release all the drug. So basically, we can see that all of them adsorb and some reversibility in the adsorption because of the release. And also we can see some conformational change as well because the frequency goes up a little bit before introducing the reducing agent. And there is a clear relationship between the charge or pegylation and adsorption. But all of them, they disintegrate and release the drug quite neatly. So by using QCM-D, you can then evaluate different kinds of candidates for nanoparticles and drug carriers to see how they behave, how efficiently they work, how they attach to the surface. And then you can draw conclusions about different design improvements that you might be interested in. Next example is about biomolecular interactions and reactions. There are many relevant scientific questions in this field in the pharmaceutical industry. A big fraction of all new pharmaceuticals today are protein-based. Proteins, when they get in touch with different surfaces, they may attach, or do you have some protein fouling basically, so you lose your valuable precious protein material. And this is of course not very favorable. You would like to find a combination or a setup where you lose as little protein molecules as possible. So here we have the questions, basically how much of a certain protein will attach to a specific surface, and can we change this by adding a surfactant or a stabilizer, and if we play around with concentration, can we change that? And is there a combination of protein surface material and surfactant that prevents the adsorption, because that is what we want eventually. So the experimental design here is that we have 2 different antibodies and we have 4 surfaces. So these materials are coated or comes on the sensor surface: gold, polystyrene, Teflon, and silicon dioxide. We have 2 different concentrations. We have excipient PS80 with and without this one in order to see how this affects the adsorption to the material. So this is the first set of results that we can see. So here we have the final adsorbed protein mass on the surface. You introduce protein solution in this QSense machine with the sensor with the specific coating, and then we can see how much actually adsorbs on each material: gold, silicon dioxide, polystyrene, and Teflon, with and without the surfactant. And we can see that in overall, the antibody 2 shows higher adsorption on all surfaces. It's highest on gold, and and but clearly lower on the other ones. And if you add a surfactant, this will clearly also reduce the amount of protein that adsorbs and binds to the surface. So it's a good idea to use this surfactant as a stabilizer. So for example, if you use a silicon dioxide surface and antibody 2, if you add the surfactant, basically nothing will stick to the surface. For antibody 1, it's polystyrene or Teflon that is a more favorable material of choice. And next, we also look into different concentrations. We have the high and the low concentration. As you might expect, of course, if you have higher concentration, more proteins will adsorb to the surface. But it is not really a linear relationship here, because after a certain threshold for some materials, you have nothing adsorbing. So if you can really fine-tune the concentration in order to minimize the fouling. For example, antibody 1 on polystyrene or Teflon, if you have the lower concentration, nothing will stick there. And for silicon dioxide, almost nothing will stick with antibody 2 for low and for high concentration. So you really need to optimize the concentration in order to get the least possible fouling in that sense. So the concluding remarks here is that it is possible to play around with different parameters here to find the material and concentration and surfactant that gives the lowest possible protein fouling on the surface. And of course, for each protein drug, you can optimize these parameters. You can also play around with temperature and other parameters in order to get the best possible setup before you scale it up, so you know what kind of transfer and container systems to use for this and that particular protein drug. Let's talk a little bit about the QSense products. We have a few different setups available, and as you have seen, apart from the instrument, the sensor is really key. Since on the sensor you can put basically any kind of material in order to mimic the situation or the experimental system that you're interested in. Apart from that, we have different softwares for data acquisition and data analysis, and we have a strong service and support team as well. And regarding the actual instrumental setups, we have first of all the initiator, which is more like an entry-level QCM-D. The QSense Explorer is more versatile system. And with this one, you can change the flow module so that you can combine QCM-D with microscopy, electrochemistry, ellipsometry, and other complementary technologies. If you would like to have a little bit higher throughput, QSense Analyzer offers 4 parallel flow modules. And the QSense Pro is a fully automated 8-channel system. So you have 8 sensors, and you can have fully unattended measurements going on. The high pressure system is an excellent tool for different kind of oil and gas and food processing related research. You can go up to 200 plus bars of pressure and 150 degrees Celsius temperature. With regard to sensors and coating materials, we have lots of different possibilities here. We have over 50 standard surfaces and coatings, and we have lots of more surfaces and materials available on demand. And we have also a strong team that can custom make sensor surfaces. If you would like to have one specific material, maybe one specific mineral composition or something like that, can create that for you in our lab. So we have a wide range of different polymers, metals, alloys, basic elements of different kinds, oxides, nitrides, glasses, steels, alloys, functionalized surfaces. So these are some examples of metals. Depending on what kind of system you would like to mimic, maybe you're looking into some new implant material, or you're working with semiconductor industry, or like new battery technology, you would like to study different surface processes or interaction processes. So we have a wide range of materials for lots of different applications. We have several polymers, we have functionalized surfaces, we have cellulose, biotin, histag, and then we have also surfaces that are tailored for cleaning research and development. We have different stains and coatings that can be studied. You can study the removal of different materials from the surface when you develop a new, like, detergent or some dishwashing powder or something like that. And here are some examples of sensors that are offered on request. These include like different mineral surfaces that can be used, for example, if you're working with mining or oil recovery. So we have lots of different options here. So the machines, the instruments, the sensors, we can combine it in many different ways and add different functions, add different materials and capabilities depending on your requirements and needs for your particular applications. So with that, I would like to thank you for your attention. If you would like to know more about this, you can visit us at our website, biolinscientific.com. Or you can also contact me directly. You can find my email address here. And thanks a lot for your attention, and I hope to be able to visit you physically next time when we are not in the corona times anymore. Thanks a lot, and take care. Bye bye.